Hello and welcome to today's GEOTOP-A lecture. We have the great pleasure to welcome Peter Bubenik from the University of Florida, who's going to give us a lecture on topological data analysis for biological images and video. Thank you very much for joining us today, Peter. I look forward to your talk.
So thank you for the introduction, and thank you for the invitation to speak here today. In today's talk, I'm going to, the main goal is to discuss two applications of topological data analysis to biological images and video, but I'm going to start with going through some of the mathematics that underlies this work, and please interrupt me at any time, or if you want to ask questions. I think there's a way to do that, and the channel is going to be monitored for any questions that are posted, and then hopefully somebody will interrupt me and ask your questions on your behalf. All right, let me switch to presentation mode here. All right. So the subject of today's talk is topological data analysis, and the goal of this subject is to analyze data that has a rich geometric structure. So we would like to quantify and learn from the shape of data. So we're particularly interested in data that has some interesting geometry that's both crucial for understanding the data and is sufficiently complicated that kind of other methods have difficulty making full use of that data. Now, the first and probably most important step is encoding the data in a way in which we can apply ideas from algebraic topology. And the main tool that I'm going to be using in today's talk is something called persistent homology, which looks at a one-parameter family of spaces and tries and kind of quantifies keeps track of homological features as they appear and disappear as this one parameter changes. Now, constructing a parameterized family of spaces from data is something that is kind of the initial step, but it's in some ways the most important step because there's a lot of flexibility in doing this, and it's really crucial to kind of think about, understand the data and understand the topology and come up with a way of quantifying or building a mathematical structure that's going to be able to capture the shape that we're interested in. So I'm just going to show the simplest way of doing this, and it's probably the most popular way because it is the simplest, but again, I encourage you to kind of think more deeply for your data about maybe other ways of encoding it into a family of spaces. So here we have a finite set of points in the plane. We can consider the union of balls of some fixed radius centered at those data points. And then as we vary that radius, we get an increasing family of spaces. And that union of balls is covered by the collection of balls. And that collection of balls has something called a nerve associated to it. So this is a combinatorial representation of that space. And for our purposes, we're going to think of it as a simplicial complex. So it consists of vertices centered at the starting data points, and then edges corresponding to intersecting pairs of balls, and then we have triangles whenever three balls intersect, and so on for higher dimensional simplices. So we see as we increase the radius, we get this increasing family of simplicial complexes, and homology is particularly good at recognizing cycles. So these are homological features in degree 1. So homology records cycles modulo boundaries And at this stage of the filtration, we have two linearly independent non-trivial cycles. And then continuing, eventually our union of balls becomes contractible, and we don't have any more non-trivial homology. All right, so mathematically we encode this as an increasing family of simplicial complexes. We apply homology with coefficients in a field, and because that is a functor,
Peter,
yes?
just one question. Somebody was asking, what data points are you putting balls around? All of them? So maybe say something about how you're understanding your data set.
So in this case, the data consisted of a finite collection of points in the plane. So we use all of those points in this simple example. Okay, thank you for that question. In homology to this increasing sequence of simplicial complexes, we get a one-parameter family of vector spaces and linear maps between them. Now, it's a remarkable fact of linear algebra that there exists a coherent set of bases for all of these vector spaces. So it's possible to choose bases for each of these vector spaces so that so each linear map is completely determined by either sending a basis element to 0 or sending a basis element to another basis element into one-to-one way. So in other words, consecutive vector spaces have bases, and they have a matching of basis elements. So we can use these matchings to define a equivalence class that gives us a partition of all of our basis elements, and the classes of this partition give us intervals. So these collection of intervals give us something that we call a barcode, and each of these barcodes corresponds to a homology class that's born at the left end of the interval and that dies at the right end of the interval. So we move on to barcodes coming from real data. We have many more of these intervals, and we have an image like the one on the left. And once we get kind of too many bars to fit on the screen, a more convenient encoding is something called a persistence diagram, which we see on the right. And this has dots whose coordinates correspond to the left and right endpoints of corresponding bars in the barcode. And one of the fundamental results of this subject is that this construction is stable. So under appropriate choices of metrics, if we perturb the data by some amount epsilon, then the barcode and persistence diagrams will also only be perturbed by epsilon. All right. Now, we really want to do data analysis. So we'd like to be able to apply tools from statistics and machine learning. Now, if we work with barcodes and persistence diagrams, it is possible to do data analysis. There are some things that one can do. However, it is maybe somewhat difficult, and the amount of statistics and machine learning we can do is somewhat limited. To be able to use kind of the full toolkit from these subjects, we would like to have a topological summary that is a vector. So we can do this if we just map our persistence diagrams into a vector space. So this map is sometimes called a feature map. There's a corresponding kernel, and kind of a more sophisticated way of looking at this is that we are mapping our persistence diagrams into a Hilbert space. Now, there's many ways to do this. I'm going to use one that I'm partial to, that I came up with, called the persistence landscape. So I'm going to start by defining this for you. So An important observation about the persistence diagram is that if we fix a parameter t and we look at the corresponding point on the diagonal and we look at the upper left quadrant from that point and we count the number of dots in the persistence diagram in that quadrant, then we recover the dimension of the vector space at that parameter number. Okay, so this is a theorem. It's probably best understood in terms of something called Möbius inversion. And this goes back to the kind of beginnings of persistent homology. Now, an important thing to keep in mind when we look at these persistence diagrams is that they're coming from experimental data that is should not be thought of as kind of a precise pair of real numbers with arbitrary accuracy. There's really some sort of error bar that should be put around these dots on the persistence diagram. And we should think about how much faith or confidence we have in this count here that we've obtained of 3. So if we're allowed to perturb those dots by a little, then perhaps our count might drop. And there's a nice mathematical way of kind of reducing our persistence diagrams to kind of increase our confidence. And there's a nice word that's been used to describe this called erosion. So in fact, the persistence diagram can be eroded in a precise way. And the effect of that also erodes the bars in the barcode, and it just kind of reduces, shrinks the bars. And for the persistence diagram, this shrinking moves the dots toward the diagonal. And so equivalently to moving the dots close to the diagonal, we can move the diagonal kind of closer to the dots. So if we do that, then our quadrant, our upper left quadrant, also moves away from the diagonal, and then our count might decrease. So one question we can ask is, how far can we move that quadrant so that the count is at least 1? So maybe the count isn't really 3, but maybe we're not so confident that the count is 3, but we can say that we're fairly confident that the count is at least 1. So for each parameter value t, and for each count, we can ask how confident can we be in the count being at least that number at that parameter value. So this gives us a 2-parameter family of numbers, or a 2-variable function. And this function is what we call the persistence landscape. So the definition is just this simple definition at the bottom here. And if we plot this kind of 2-parameter family, which we can think of as a sequence of functions, and we graph those functions, this is what they look like. So it's a decreasing family of piecewise linear curves, and under some mild hypotheses, these are square integrable functions. So they live in a Hilbert space. So there's this simple classical linear product, and in fact we can discretize these, which is convenient computationally. So just chop up our parameters into some grid, evaluate each of the persistence landscapes, concatenate these numbers, and now we're either in a function space or a Euclidean space, and we can do essentially all of linear algebra, statistics, and machine learning. All right, a few theoretical properties that are reasons that I like this construction. So as I said, these are piecewise linear functions with slope 1. Because of that, they have very simple derivatives. And this actually ends up being very convenient if we want to combine persistence landscapes with deep learning algorithms, because learning uses stochastic gradient descent, and having simple partial derivatives is very convenient. The construction, again, is stable. It's also lossless in the sense that we can recover the persistence diagram from the persistence landscapes. And one crucial property is that it doesn't involve any parameters. So I think this is maybe one of the only kernels that I know of that doesn't have a choice of bandwidth that's required to use it. Something a little more sophisticated is that it's injective on certain generic empirical measures. So not only can you recover one persistence diagram from a persistence landscape, if you have an average of a number of persistence landscapes, you can recover the persistence diagrams that went into it. And then the most recent result at the bottom is that there's a nice combinatorial construction of the persistence landscape. So the persistence diagram can be obtained from the rank function using Möbius inversion. And if we take a graded version of the rank function, then we apply Möbius inversion to that, then we obtain the persistence landscape. All right, so that's the end of the background theory. So this actually might be a good spot to take questions. Are there any other questions on the theory part of the talk?
I don't see any other questions on the chat, no.
Sorry, Catherine, was that a no?
That was a no.
No, okay, all right. So again, please interrupt me as we go along if there's any other questions. All right, so the first project that I want to talk about...
Sorry, Peter, there is a question.
Yes.
Could you explain barcodes one more time, how you get them from homology, please?
Yes, I'd be very happy to. All right, so we have a family of vector spaces and linear maps between them. And in the computational setting, all of the vector spaces are finite dimensional, and all of the linear maps are represented by matrices. And so if we change bases, then of course the matrices will change. And it turns out that if you have two vector spaces and a linear map between them, you can represent that by a basis by a matrix that consists of zeros and ones, where all the ones are on the diagonal. Now, kind of more than that, we can do it at every step of the way, so that each of the basis elements is either being sent in a one-to-way to another basis element, or it's being set to zero. So we can kind of represent this with a graph, with the bipartite matching. So the basis elements that are matched are connected by an edge, and if a basis element goes to zero, then there's no edge kind of to the right of it. And if a basis element kind of appears, then there's no edge to the left of it. And then by concatenating all these edges, then we get the barcode at the bottom. All right. And of course, this can be thought of, this is a central theorem. It has different interpretations. It kind of at the high level, this is a result of a theorem of Gabriel about representation theory of quivers. It also comes from kind of a graded version of modules over PID, and then from a more computational point of view, there's actually an algorithm where this just drops out, and a lot of the mathematics that I've talked about is just implicit. The vector spaces kind of never appear, and everything is just done by a matrix reduction algorithm. Okay. So the first bio application is to images of cells. So these are images taken using microscopes, light microscopy, and biologists are very interested in something called the actin cytoskeleton. So this is kind of a
screen, Peter. Sorry, Peter.
yeah. I'm about to switch
Okay.
to another image, so I'm not going to do that right now. This actin cytoskeleton is kind of a scaffolding for the cell, and so it's kind of the dark image. And however, an important part of this structure is that it's dynamic. So I'm going to switch to a video here where, again, videos may be a little tougher for everybody to see, but if we stay with this, hopefully you'll get the video moving on your screen. So these actin polymers grow on one end and then degrade on the other. And because of this, the cell actually uses them to move. So not only do they kind of give it its structure, it's also important for cell motility and for things like muscle contraction. And because it kind of helps the cell move, it's important in wound healing. And so it kind of has a host of applications kind of in biology and medicine. And people are really interested in understanding this structure in the cell. So when we started talking to our biology collaborators, of course, these things have an interesting shape. So it was kind of a natural place where we thought that we could make a contribution as applied topologists. All right, let me go back to my talk here. And let's go to full screen. All right, there we go. Okay, now one thing that was nice about this project is it was challenging because the images were very high resolution. So we have on the order of 4 million pixels in our image. And the features that we're looking at are small scale that we only really see at the full resolution of the image. So kind of downsampling would lose the structure of interest. So the solution that we came up with was to subsample patches of our image. So in step 1 here, we see we've extracted a small part of our image, so a little disc-shaped patch. And these are grayscale images, and we convert them into a collection of points by now sampling the patch, so sampling the highest intensity pixels, giving us a point cloud. And now again, homology, the thing that it's particularly good at is detecting cycles. However, in this, for this data, the interesting features are these linear features. Now, there is a version of homology that's kind of good at detecting local linear structure, which is called local homology. So we used that as an inspiration for a kind of quantitative version of local homology where we introduce a collection of points along the boundary of the disk that we're looking at. So this is step 2. So we have your sampled points in the interior of the disk together with a bunch of points around the boundary of that disk. And now we construct a increasing family of simplicial complexes from that point cloud, just like we did at the beginning of the talk. Now, actually, we do something a little more sophisticated than the check complex construction. We do something called the Delaunay complex, also called the alpha complex. And this complex actually is produces a persistence module that's isomorphic to the one produced by the Czech complex. So the persistent homology ends up being the same. The advantage is that it has far fewer simplices, so computationally it's more efficient. And in 2 dimensions, it can be computed very quickly. So the steps of this one parameter family of simplicial complexes we see in steps 2, 3, 4, 5, and 6. So at 6, we get the Delaunay triangulation of the disk from this collection of points. And now we compute persistent homology. We get the persistence diagram that we see here at step 7. And I've highlighted the 4 most persistent dots in the persistence diagram. And as part of the algorithm, we can look at representative cycles for these persistent homology classes. And in step 4, these are indicated and colored in the same color as the corresponding dots. So you see here there's 4 kind of representative cycles that pick out 4 of the regions in this patch that kind of pick out this X-shaped structure. And then at the end, we encode this as a persistent landscape, and that gives us a vector representation. All right, now we repeat this construction for many patches that cover the image. And the first thing that we did was something very simple, which is something called, an example of something called unsupervised learning. So we have a moderate number of images. We have a large number of patches for each image. For each of those, we get a vector from the persistence landscape. So we have a large collection of points in some very high dimensional space. And we did something very simple, which is just project down to 1 dimension. And what this does is it gives us a real number associated to each patch. Call that real number a score. And then for each pixel is covered by some number of patches. We get the average score of those patches. And we can use this to visualize the images. And so the projection that we used was something called t-SNE, which is usually used to project high dimensional data to 2 dimensions, but it can also be used to project to 1 dimension. On the left here is a histogram of the scores of the patches coming from the image of the right. And then we also get a visualization. So this is already interesting, and we see kind of that at the one end of the scale, we have patches coming from the outer boundary, and this is where we see the kind of linear structure of the actin filaments. And the actin is also found in the interior of the cell, but has kind of a more point-like structure. So this is already kind of an interesting visualization method. But what we're most interested in is doing classification and regression. So this is a version of something called supervised learning. So in this case, we have an experiment. So we have kind of 2 classes of data. We have the control, and then we have, say, a mutant where maybe some gene has been changed. And we split the data into training data and testing data. So the training data has been labeled, and so each of the patches gets a label. Each of the persistence landscapes gets a label. We take those labels to be either -1 or +1, and then we do something called a support vector machine. And the particular version of support vector machines that we use is something called support vector regression. So the idea is we have this high dimensional point cloud consisting of 2 classes, +1 and -1. We look for a separating hyperplane that puts the +1s on one side and the -1s on another side. It ends up being an optimization problem where we have a penalty given by kind of the distance of points to the separating hyperplane, and it ends up being a least squares problem, so it can be computed very quickly. And we produce a model which consists of some hyperplane together with a normal vector, and then the score that we get for some test data consists of the distance to that hyperplane kind of scaled by the normal vector. All right, so for our test images, each of the patches, we look at its persistent homology, look at its persistence landscape, take the appropriate dot product. That gives us a score, and then for each pixel, we take the average of the scores of the patches containing it. All right, so this is what happens. So here we have two cells, control cell on the left. On the right, we have our perturbation, experimental perturbation, which in this case, our collaborators have knocked out a certain gene, PFN1, that encodes a protein called profilin that regulates actin. So in this case, kind of this is a very strong perturbation. To see by the naked eye that these cells are different. And the interesting thing that two things we get out of it. One of which is that we do get a good classifier. So at the top, we have the histogram of the values of the patches of these images, and the average of those patch values, or average of those scores, is indicated by the dashed line. So on the left cell, we have a negative score, so we assign it control, which is correct. On the right cell, we get a positive average, so we assign it that class, so we get the correct classification. So in this case, we got 100% classification, which is great. However, what's I think most interesting thing about one of our most interesting contributions is that we get a visualization of what parts of the cell are responsible for that classification. And our biology collaborators found this part to be interesting and surprising, but something that they could interpret. So, in fact, the part of the cell that was used by the classifier was not on the leading edge. On the image on the left, we see that the parts that give it the strongest score are kind of not on the very leading edge of the cell where we see the actin filaments most strongly, but a little bit further in. So the biologists found this useful for kind of suggesting hypotheses for further experiments. All right, so that brings me to the end of the first application. I have one other one to show you. So quick questions, we can maybe take them now, otherwise there'll be time for kind of questions for both applications at the end. Were there any questions in the chat?
So there's a question saying, how do we go from step 1 to step 2 in the first example where we got a collection of points from the image patch?
Step 1 to step 2. All right, I'm going to assume it's maybe it's the labeling that I used here.
I think it's the labeling, yes.
Okay. Yes. Okay. So here there were some decisions that needed to be made. So first of all, one thing that we didn't do which is just to use the grayscale values of the pixels to give us a filtration of that cubical complex. that maybe seems like the obvious thing to do, but there's some difficulties with that. If you end up with one pixel that has a very different grayscale value from its neighboring pixels, you end up with a long bar or a very persistent dot in the persistent homology. And these kind of one-off pixels are something that might just be an experimental artifact due to noise. And to get rid of that, you need to do some smoothing, but if you start smoothing, then it's very easy to lose the linear structures that were of interest. So because of that, we decided we really wanted to just take the pixels of highest intensity. So that's essentially what we did. So we sampled some percentage of the highest intensity pixels, and then with the additional requirement that if we'd already picked one pixel, then we wouldn't pick any of its neighboring pixels, for example, or pixels that are within some small distance. Okay, so that's some technical detail, so thank you for asking about that. All right, for the last application, actually, let me, so for the last application, we looked at videos of an organism that's very well studied by biologists. So this is C. elegans. So it's a nematode, and we'll call it a worm. So it's a very small organism, but not so small that you can't see it. And it's well studied for a number of reasons. One of which is that it's very happy to spend its life in a lab under lab conditions. So you can get an agar plate and let it crawl around on this plate. You can kind of poke and prod it by hand. And furthermore, it's kind of translucent, so you can see its insides, which is useful for doing like neuroscience. You can light it up and actually kind of see neurons firing and such things. So in this project, we were interested in studying the locomotion of these worms. So here I have kind of one of the input videos that we studied. And so there's, just to give you a sense of how this was, where this video came from. So there's a kind of a plate sitting on some bench, and then there's a video camera that's tracking this worm kind of automatically. So this is, of course, scaled up so they can observe lots and lots of these worms over long periods of time. And now, of course, we want to quantify these videos. So the initial important step was done by our biology collaborators, which is to turn these videos into kind of a succinct vector representation. So what did they do? So they extract, for each of the frames in this video, they extract a midline of this worm. So it's a one-dimensional piecewise linear curve that's kind of in the center of this worm going from head to tail. And I think they chose 100 segments, so it's kind of 100 curves, 100 piecewise linear curves, so kind of we have an angle from one segment to the next one, so roughly we can describe it with a vector in R100. So that's the data that we were given as mathematicians to work with. Now, before I go on to the analysis, I want to look at, well, first of all, I should say if you look at the paper, there's a number of things that we did. We looked at kind of lots of these videos. We did some similar things that I did in the other project. We did kind of classification and regression, and we're able to kind of say what kind of environment the worm was moving in by looking at its locomotion, by looking at the TDA of its locomotion. Now, for today's talk, I'm going to look at just this one video and try to look at it in detail and correlate kind of the homology to things that are seen in the video. So I'm going to talk you through this video and kind of highlight things that hopefully later on we're going to see in the persistent homology. So in this video, it's 21 seconds long. Initially, the worm is moving forwards in this sinusoidal motion. At some point around 9 seconds, it changes its mind and transitions to moving backward. And the backward motion is actually has these kind of deeper body bends. So let's look at this again. So forward has this kind of sinusoidal motion. Around 9 seconds, it transitions to moving backward in these deeper body bends. And around 15 seconds, it pauses briefly for 1 or 2 seconds, and then it continues moving backward. All right, and since I know video doesn't work so well over a lot of people's internet connections, let me do it one last time. So we have the worm moving forward, and then it transitions to moving backward. And then here it pauses, and then it returns to its backward motion. All right, okay. Okay, I'm going to switch to my tablet here, so please bear with me for a few seconds while we try to do this. Okay. All right, so starting with the video, we produced this sequence or time series of piecewise linear curves. So we have something like 20 frames a second or so. So we have our 20-second video here is now consisting of roughly 400 piecewise linear curves. So each of those curves is one vector or one point in 100 dimensions. And now we do something which is now kind of the standard method in TDA for point cloud data, something called the sliding window embedding, which is kind of inspired by Tawkin's embedding and was really developed by Jose Perea in his PhD work. And what we do is we take a window of frames of this video, and we concatenate the vectors for that window of frames to give us one point in an even higher dimensional space. And then we think of sliding that frame of windows across the whole time series, across the whole video. And for each of those windows, we concatenate all the corresponding vectors to give us one high dimensional point. And so now we're in a very high dimensional space, and we can visualize what that point cloud looks like by projecting down to low dimensions by something like principal component analysis, PCA. So on the right here, I have two PCA projections of the high dimensional point cloud corresponding to this video. So this is the projection onto the first two principal components, and this is the projection onto the first and the third principal component. Okay, so these are both views of the same high dimensional point cloud coming from this particular video. And now, since the time series is ordered, this point cloud actually has an order, and it's colored going from dark to light here. So here the dark part is the initial sinusoidal motion, which is kind of given by this cycle here and this smaller looking cycle in the other projection. And then toward the end here we have which was the backward motion, is this part here. And interestingly, already looking at the point cloud, we see here a little loop kind of corresponding to the stutter step or the pause that we had in the video. All right, so let's take persistent homology of this high dimensional point cloud. So we get the persistence diagram on the left here and its corresponding persistence landscape. And what we see is that there's some number of dots here that are most persistent, so furthest away from the diagonal. So let's count 1, 2, 3, 4, somewhat arbitrarily, are the most persistent dots on this diagram, and they kind of correspond to these peaks in the persistence landscape. And now, if we dig a little deeper, and we use our persistence algorithm not only to give us the persistence diagram, but representative cycles, we can look at what those representative cycles are. Of course, they're in this high dimensional point cloud, but let's project them down using our PCA projection. And they're the red collections of edges. And so each of those corresponds, each of the vertices in those cycles corresponds to some window. We can look at the video and look at the corresponding, like what's happening in the video. And so what turns out to happen is that the most persistent dot, so this one here corresponds to this cycle here. So this, notice, was the backward motion, and it's, I think, the most persistent because it has kind of the deepest, these deep body bends give us kind of the most dramatic cyclical behavior in the image. And the forward motion is this part here at the early part of the video. That gives us that dot. The transition is this cycle here, which, notice, kind of has this edge here which kind of jumps from one part of the video to the other. Because of that, it's born late, so it has a large birth value, so it's kind of is happening at this part of the persistence diagram. And then this pause here is this part of the video. Okay, so the last thing that I want to show you is something that I'm very excited about, is that using these representative cycles...
Peter, I have a question.
Yes, please.
Well, the question is, are you computing the persistent homology in this case? Is the one-dimensional persistent homology, I imagine, and it is for the projection or the actual original point cloud? Great question.
You should always work with the high-dimensional point cloud. Okay, the projection is only for visualization purposes. So we are, so to do persistent homology efficiently with high-dimensional point clouds, we are doing the Vietoris-Rips construction, which coincides with the check construction for vertices and edges, And then for triangles, we just stick in a triangle whenever we have the three edges in the of the triangle. And it turns out that there are very fast algorithms and software for doing this.
Okay. Well, thank you very much.
You're welcome. Great question. All right. So the last thing that I want to show you, and this is something I'm excited about, is that we now use the representative cycles. Remember, each of these cycles corresponds to some window of frames in our video. So we're able to go back to those frames and now stitch them together using the representative cycles. And doing this, we get synthetic videos that now visualize the persistent homology. So the most persistent dots in the persistence diagrams can be visualized by synthetic videos. So this is somehow a decomposition of the video using persistent homology. And we get these kind of synthetic videos that give us the characteristic motions of the characteristic movements of the worms. So on the left, we have this sinusoidal forward motion. We have this transition between forward and backward motion. We have the backward motion with the deep body bends. And then this last thing is this kind of stutter step, which is the pause in the backward motion. All right, so I'm going to end my talk by there's two papers corresponding to these two works. They've been published. There's also software that's publicly available, so please take advantage of the work that's been done. Hopefully you could use these in your own projects. And let me end by thanking the wonderful group of students and postdocs who worked on these projects and are currently working on related projects. And thank you very much.
Thank you, Peter. Very nice. Are there any more questions? If so, please feel free to unmute yourself and ask. Maybe I'll ask one to get started. So are there any other sorts of videos that you're in the process of analyzing, or are you trying to do more about nematodes?
Great question. So yes is the answer. So our biology collaborator on that project is Hong Lu. So she's at Georgia Tech. So she's a biologist. Actually, her training is in engineering. Her expertise is in microfluidics. So they have these great devices that her lab produces that are able to generate this data at large scales and large quantities. And actually, the thing that she's most interested in is the brains of these nematodes. So one of the primary reasons that C. elegans is studied to understand brains. So these organisms, they have 302 neurons. We kind of have a complete list of the neurons and a reasonable understanding of the connectome as well. And because they're translucent, if you do the right tricks, you can actually visualize the neurons firing. And so there's videos that her lab produces of the individual neurons firing. And the thing that we tried doing initially, or we wanted to do, was to look at those videos and to use TDA to say something about kind of organizing and quantifying the shape of the brain thinking and responding to stimuli and learning from responses to stimuli. So, however, when we started this project, we weren't able to do it. Kind of what they could do in the lab kind of wasn't good enough for the analysis that we were able to do. So we switched to something easier, which was to look at these videos, and we were able to do something interesting. But now, a few years later, now that we've kind of had that experience, and also the amount that they can do in their lab has improved, and so the current project is actually to go back to that. And so we're currently looking at videos of these worms thinking, and looking at the individual neurons firing, looking at the shape of those kind of activations of the individual neurons, quantifying that with TDA, and then we're hoping to kind of look at kind of globally, like the whole brain dynamics of these things. So this is, I think, a great object of study, and of course there's kind of similar model organisms, and people also are looking at synthetic versions of this in the Blue Brain project that I know you've been involved with. And I think there's a lot to be done, and the technology is improving both on the biology end and on the TDA end. So I'm hopeful that kind of we're going to be able to do more and more of in this direction in the future.
Thank you, Peter. Very interesting. I look forward to seeing the results. So there's one more question in the chat that said, in the third application, if this person understands correctly, the token embedding or something similar was used to create the high dimensional point cloud. Just asking for confirmation that it's basically a tokens embedding.
Yes. Actually, let me go back to that. Let me see if I can get back to that video. Yeah, so there is a choice that needs to be made there, which is the size of the window. So the other part of the project that I didn't talk about is we used the TDA coming out of these videos to do some classification, be able to say something about the environment that the worm was moving from, and just from looking at the videos. And initially we just kind of picked a certain window length, I think maybe 30. And that's the one that I have in this visualization on the right. But then we tried other window lengths, and the results were very similar. And in fact, I think we tried all the window lengths between 1 and 30, and to see which one was best. And the interesting thing is that the one that had the highest classification accuracy was a window length of 1. So in fact, kind of not doing a sliding window, just using each frame as its own point cloud. The machine was actually kind of happiest with that. But us as humans, we weren't so happy with that, because we got an image that looked similar to the ones on the right here, but were actually not nearly as smooth. They were a lot more jagged and noisy. Because when we introduced this sliding window, I mean, we're not really averaging, but effectively we're amalgamating neighboring data points. So it ends up kind of, in some sense, smoothing the data. So visually it was nicer and more helpful for us as human mathematicians doing the data. But well, the one answer is that the window length ended up really not mattering, and the machine was slightly happier, just not even using a window and being just one frame at a time.
Interesting. Okay, 2 more questions. One more question, rather. I see that the persistent diagram is created for one video. Did you try other videos? In the research, you got similar persistent diagrams. Is there any idea of the distribution of points on the persistent diagrams?
Yeah, so that's a great question. So here we have one from one kind of short video. So in the more kind of machine learning part of the paper, we looked at longer videos. So we got more complex diagrams. We looked at more videos, and really we'd like to, and in the labs they can generate hours and hours of these videos, and let me give you my vision of what I would like in an ideal world, and if I had kind of more people interested in doing this project with more data and trying to do different things with it, is that kind of each worm you might track for a day or maybe even for its whole lifetime, which isn't that long, a couple weeks. And then maybe we look at snippets of its movement, maybe 1 minute at a time. Each minute has a persistence landscape, some high dimensional vector, some sequence of persistence landscapes with the high dimensional embedding, some point cloud associated. Things that we're studying don't really need 10,000 variables to explain it. That these worms really can be described fairly well using lower dimensional models. So this high dimensional data lives on some low dimensional manifold maybe, or stratified manifold in this high dimensional space. And then the worm, as it's aging, is maybe moving on this manifold. And then also its kind of day-to-day business is also moving in the manifold. It might be moving more quickly at some point. At other points, it might just be sleeping or resting. And by using topology to understand this manifold, we would get some kind of representation of the space that we're studying in that would be useful to the biologists. So I think this point of view is useful, and there's lots of people in TDA, I think, kind of trying to do this, to construct kind of models for biological and other systems that aren't derived from kind of PDE models in the way applied mathematics is often done, but in kind of a more data-driven way.
Okay. Thank you very much, Peter, for your answers to the questions and for a really interesting talk. Thanks to all the participants for having joined us today, and look forward to seeing you next time. Thanks a lot.